Okay, my friends, I have been working with some people over in North Ireland that are doing a study of bacteria that's within the body. And the bacteria in the body are the chemists that provide the molecules using enzymes. We're going to go through this, but there's some new research that indicates that if we can get that bacteria back doing its job in us, and get these enzymes to break down molecules the way they should, there's a chance we might be able to help with some of these diseases that just never seem to go away. They just get worse and worse and worse. And it appears to be now that there may be some interaction between the bacteria, the enzymes, and the end result, which is invasion and disease. Because these enzymes live in a certain layer in the body. Now we're going to get into it, and we're going to go. All of these are different research papers and articles and all that. Again, I'm working with the people over in Northern Ireland, and they have started to become involved and start. Not deeply yet, but we're hoping for that. And I want to show you why I think this would be a good thing to do. I'm not giving any medical advice whatsoever. This is 100% strictly research. And it's going quite well. And we have a staff of people that are fully trained on this. We triple PhD for bacterial engineering. Marguerite went to Harvard for ribosomes. I've gone to Johns Hopkins for genomics. We all have some fairly decent skills to be able to address this. But it goes a little bit contrary to what the current thinking is. So again, research only. There's no medical advice here whatsoever. What we need to do is develop a database so that we can determine what the medical advice should be. Right now, there's no database. We don't know how the human body works. It's unknown. And I'll show you how it works, and it works because of bacterial enzymes. That's what we need to control. Okay, this is a lot of information here. Each one of these is an article from either the National Institute of Health or Harvard or Johns Hopkins or wherever. And these are all from Yale, from National Institute of Health. This is all serious stuff, National Institute of Health. This is all things that they're looking into, and they have not concluded their research, let's put it that way. They have been just typically talking around these things. I want to show you all of this in one consolidated presentation so that you can understand how deeply associated these things are. This is the last thing right here. In a global research effort, scientists have uncovered a relationship between metabolism, that means the way your body breaks down foods and produces things, the metabolism problems in the brain, and a range of psychiatric and neurodegenerative disorders from autism to Alzheimer's. Now, my approach to this is that everything is coated by a membrane, and the membranes fail. They call it tight junction membranes. And if they lose that tightness of the junction, they're invadable, and molecules move in and out freely. And then you get all kinds of crazy things because you're getting short circuits. That's one of the things that I believe is part of this. Now, what we need is a database to find out what bacteria is creating those specific enzymes that create that specific chemistry. All right, this gets a little bit involved, but it's not bad. And even if you have good genes, listen to this now. The genes that are in you, the genetic material that says, all right, I want 5 fingers, I want 5 toes, I want 2 eyes, and all that stuff is built into this program code. And it also says, here's how we react to this, and here's how we react to that, and here's what we do about this. And even your emotions, I believe, are built into these codes. That's why you get traumatic injuries in your DNA, literally, even though it's just you see something terrible, it haunts you possibly for life because it's locked into that code in your DNA. Your DNA is not junk DNA that they say, oh, it's all junk DNA, 98% of it doesn't do anything. No, that's your memory, that's your immune system taking hold and starting to learn. That's your emotions. To me, that's what that is. Now, your genes, they have to turn on to work. So your gene sequence is there. It's like a program. But if you don't have the button to push to make the program run, the program never runs. So what happens is the only way you can push that button is with the enzyme. The enzyme pushes the button. There's no other way to do it. The gene can be working perfect. Nobody pushes the button, it never turns on. You don't get the product. So typically the group, this is called methylation when they push the button. Methylation, they methylate the DNA and literally turn it on. Now this is not my words, this is in quotations. Typically this group is added to specific places on the DNA, this methylation, where it blocks proteins that attach to DNA to read the program, to read them. The chemical group can be removed through a process called demethylation. Typically, methylation turns genes off and demethylation turns genes on. All of these little things we're going to go through. I'm going to go through them quick because we're starting to get into the research, but people have to understand how involved your bacteria is in your body. And there's been a new development in 2018. They discovered how these things move through the body, through this fluid-filled highway. And we're going to be going through that. We'll start with that, and then we'll go on from here. But each one of these we're going to address quickly. Boom, boom, boom. And there's a ton of them. All right, before we start, you have to understand, this is brand new. We're going to be talking about the interstitium primarily, which is a new organ system, really. It's a network of fluid-filled cavities found everywhere in your body, everywhere. Your body is lined with a network of fluid-filled cavities that until now were unknown to science. They didn't even know about this, and they just reported on it in 2018. Now, I discovered this and reported on it in 2015. I wrote a paper about it, this fluid-filled network highway. It involves one gigantic system that covers the whole body, and all of these lymph fluids really is what's flowing through here. And they still don't understand this because this is pretty brand new stuff. And all of the stuff I'm going to talk to you about today is based on this fluid-filled highway and the enzymes and bacteria that live in there. And I'm going to actually show you them living in there because now they can actually see them. Okay, this is 2018 is when they announced this. Scientists say they found a new human organ, dubbed the interstitium. A new organ is a network of fluid-filled cavities everywhere in your body. And they never knew about this because they weren't visible. By the time they autopsied people, the fluid was flat like a pancake, and they thought it was a thick layer. All right, so this is brand new. Okay, this is blowing my own horn, but this is true. In 2015, I discovered this layer, and it was fascia, and at that time they called it fuzz. It had no biological duty whatsoever. It just was laying there like padding. Now, I said all fascia is made of collagens. Now, this is important to remember because there's 20 different types. Collagen is the fabric creating fiber and elastins in a sheet that wraps everything individually. This is this fluid-filled highway. And then comes together as one giant system. The whole thing comes together. It's all one big fluid-filled highway. Now listen, this is really important. There are over 20 types of each collagen and keratin. These are the chemicals that create these webbing and these fibers and these juices. All right, some protect from acids. All right, some of these different collagens and keratins. Some protect from like salts and caustic things. Others mineralize for strength. They give it strength and stretchability to give you tight junctions. And the variety is amazing. It's just amazing. these fibers are infused with silicon. This is my statement from 2015. One giant system. It's a complete fascia, protects every bit of your body from invasion, including a complete fabric bag, your skin. Your skin is not just one type of fabric either. You have tough skin, fine skin, all that, and I go through the whole thing. All right, just so you understand, a fascia is a tubular, fluid-filled fabric network. Instantly reconfigures itself. If you damage it, you pull it, you stretch it, you're doing all the time, but it puts itself right back together. And I was working with people over in Germany and all those places, and at this time they thought it was fuzz. They were talking about just being fuzz. And the only people that knew anything about it were sports injury people, because you get injuries in these membranes, and they pull and they stretch and they rip and all that. And I'm going to show you in a second. I can prove that as well. So anyway, I understood this, and I also knew that it was associated to acupuncture points where these things invest in each other. I'm going to show you some of that happening right now, just to prove myself, so we can go on from here. Okay, the reason I'm showing you this is how I discovered the interstitium and these fluid-filled highways. Now, this is a rock, but it is what I call a mud fossil, and that's what I discovered mud fossils in 2010 or so, and realized these were all body parts. And then I started to really research them deeply and get into the actual chemistry and the anatomy really, really deeply. I mean, as I will show you, and I discovered things nobody's ever seen before, because I can see them preserved in their exact entirety. Why they missed the interstitium was because by the time they looked at it, it had already dried out and turned flat. All the fluids went away. They never saw it. Up until just 2018, they agreed, yes, Roger was right. Because I kept saying, this is all different than you guys are talking about. So anyway, here's the deal. This is the flap that is that flap. That connects every organ and every body part to the next body part. And there's two little tabs in here, two little holes that appear to be the connection for the fluids. Now, as I had in my paper, some organs are filled with acids. Your stomach is just a bag of acid. Your pancreas, your gallbladder, your liver, your kidneys, your heart, your lungs, they all have a specific chemistry that is only created by bacteria, which are the enzymes that live in this fluid-filled layer that is the interstitium that I discovered in 2015. Nobody knew about it. They still don't even, most doctors still have no clue about it. This thing travels very slowly through the medical community. If they went to school 10 years ago, they're not interested in, well, I shouldn't say that. I don't see much interest, let me put it that way. Well, I see no interest at all. So let's just be honest. Now, this, I am going to show you how this transfers its fluids from this organ, or in this case a chunk of a muscle, to wherever it has to go to, because this muscle is going to be protected by, let's say, something that has more saltiness to it than acids. But it may be connected right up to your stomach, because something's going to be connected to your stomach. Your stomach has got all kind of acid in there. It's got to be a different chemistry. That chemistry is different chemistry. So these two little tabs that come out that connect to the next set have to have a different chemistry here, and somehow it isolates from this one to the next one, because if this was protecting you from blood, protecting your lung, and it's right next to your kidney or something, you can't have the same fluids here in that membrane as you do around the kidney. It's just not going to work. Or same thing around the stomach. It's not going to work. They have to be different chemistry. So I'm going to show you the little holes that connect this. And now they don't know that this is a new organ. This is another new organ that's unknown to science right now. And I know that for a fact because I've talked to some of the top people in the world about this, and they don't know that that is a special type of organ right here, which is the attachment. It's completely different from the rest of that tissue. It doesn't look it here, but I'm going to show you in the microscope in my mud fossils that it is absolutely no question it's a different piece of material. Now, I'm going to look in the microscope and show you these two dots. What am I talking about? They're right down here. You can't see it in this picture, but I have it up in the microscope right here now. So we're going to be looking right down in this area right here. And there it is. All right, take your time and look. See those two little dots right in the center there? That one and that one. That tab comes down and it would lock into another piece of body part. And these, I believe, jump over into that other part. I believe that somehow the fluid goes in and comes back. I'm not exactly sure about that. Nobody knows even about it. So it's totally unknown at this point. However, that is, I believe, how it transfers the fluids between one organ and the next organ. That's all I can determine because they have to be different chemistry. All right, what you have to understand is that every part of your body has a different chemistry. You've got your esophagus, you got your stomach, you got your small intestines, you got your large intestines, you got your rectum. All of that food passes through there. It's going all right from the beginning to the end, through your saliva, through your stomach, through everything. So every one of these is open to invasion. You're going to eat all kinds of stuff that's not going to be good for you, and it's going to have to go through you, or it's going to have to be dealt with you by something in your body. And the only thing that creates something fast enough to deal with these invasions is enzymes, and the only thing that creates an enzyme is a bacteria. So let's remember, bacteria is the focal point of this whole research project, is to find out what is a normal, healthy bacterial community in what parts of the body. This is kind of a daunting task, but we have to come up with novel ways to investigate what's coming out of here. And the whole thing, literally, once it goes through your body and comes out as poop, is really basically what you're made of. That's kind of crazy sounding, but it's true. You're made of bacteria, literally. All of this is nothing more than a factory for the bacteria to produce the chemistry that uses all the things that come through your factory and sends them off over to the lungs, over to the kidneys, over to the heart, over to muscles and tendons and toenails, and whatever it needs. But the only way that can convert that stuff is bacteria. All right, I hope that makes sense to you, because that's the key right there. And they live in these layers. And again, they never knew this. All of these have the same layers. But at this point when this book was written, they didn't realize that the interstitium was a fluid-filled highway. Now, I've read this book front to back, and it's as detailed as any medical book there is. And in the beginning, when a baby's born, they have no digestive systems to speak of. It says the digestive system of a newborn, unlike the adult body, the fetal digestive system does not have a digestive function from birth. There's no digestive function, but it's ready to program from local bacteria, which is vaginal juices, fecal matter, and all that. When they're born, all of that stuff should get all over them. And they're trying to keep them too clean now. See, I shouldn't say that. Forget that. Keep them clean, do whatever you want. But I'm telling you, if they don't have the right bacteria, they're not going to stay healthy. All right, so they do not have any digestive function from birth. The stomach and intestines are reduced in size, while the liver is huge, much more developed. Look at the size of the liver. And what does the liver do? It deals with toxins. So in the beginning, the baby can't deal with the toxins. It's got to get some programming. How does that programming happen? It happens by bacteria creating enzymes. The enzymes creating these little messenger RNA packages which program your DNA to be able to recognize and deal with invaders. And I'm going to show you a body actually now recognizing and dealing with invaders. So this goes on. There's some serious stuff. You know, there's islets of Langerhans, and there's all kinds of chemistry going on. But it's only chemistry can only happen by bacteria. So let's remember that. And again, this is not medical advice. This is medical research. All right, so we know there's a fluid-filled highway underneath the membrane. So the membrane protects this muscle from invasion by anything that's out there that's not supposed to get into it. Now, the fluid-filled highway is right underneath the membrane, and here's what it will look like right here. All right, this is right from 2018, Meet Your Interstitium, a newfound organ, and here it is. Now, the white layer is this newfound organ. That's the fluid-filled highway. It's exactly what it says. You see, scientists discovered new organ consists of fluid-filled spaces. It's a fluid-filled highway right here. All right. This was known before, the black layer, this dark layer, and with all these little holes in them. That's really your white blood cells, lymphocytes and so forth. And the bacteria and enzymes live in those. And they create the enzymes and they create all the products they need. And then they grab them and shoot out into the fluid-filled highway where little flagellas zip, 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 all over the whole body. Because it's continuously one big, huge body. And they agree with this now. It's all built together. It's a newfound organ. The whole thing is one organ. It's the biggest organ in the body. I never knew it existed. And the doctors still don't understand it. And I ask a lot of them about enzymes and probacteria and, you know, what about this particular layer? They don't know about the layer, first of all. And secondly, you know, that stuff, yeah. You know, we got other better ways to do it. That's on the back burner. You know, really very little interest. So we're hoping to get a database, and then if we can establish relationships between missing bacteria and diseases, well, then it's all over. All right, you see this? Your textbooks are wrong. This is what cells actually look like. Now this is GoCall and it's Seeker, and I posted back here 3 years ago. We could literally save lives if we can get bacteria in this interstitium layer. He's going to actually show the layer. He's going to show the activity of the bacteria collecting up the ribosomes to go deliver them. The ribosomes are the enzymes. Now watch what happens. I'm going to only play this for a minute or two.
I still remember the moment.
Hold on, let me turn this up.
I will never forget. The hair on my hands just stood up. It's a microscopic universe within each cell.
This is an unprecedented view of the cellular world, where we can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
Focusing only on the crawling immune cells, we've noticed two classes of them. One team was not hungry at all, but it was very active in terms of trying to figure out, what the environment is. There was another one that was kind of slouching around with a lot of food in its belly. We can actually conceptualize and visualize and analyze the contents of each of these cellular compartments in this crawling immune cell as it's kind of, scooping up its environment. I mean, that is a level of details no one's seen before.
We're living in a new era of cell.
Let me just explain this to you. All right, let's start from the beginning where we're seeing this right here. This is one of these lymphocyte cells, these white blood cells. And these, I believe, are all of the ribosomes, which are the enzymes. And they're in a ball form. This guy here is coming running around here, grabbing, scooping them all up, and then he goes right out from the cellular layer into the fluid-filled highway to deliver. Now they say he's scooping up sugars. Well, I say he's scooping up enzymes. They don't know what he's scooping up. But
we see immune cells scooping up sugars in the ear of a zebrafish in real time.
Focusing.
You see him go straight out, and then now he talks about two types of cells, two types of immune cells. Which
only on the crawling immune cells, we've noticed two classes of them. One team was not hungry at all, but it was very active in terms of trying to figure out,
Let me talk to you about something about this. This is a magnetic chain of polypeptide proteins. All right, it goes searching around, and it would see something with this particular signature. It says, "Uh-oh, that guy's no good." It reports back to the bacteria, which they think it's eating something. It's gobbling up all those ribosomes to go deliver them. That's my thinking at this point. But this is the messenger. Now, remember, he says they don't do anything. They don't eat anything. This one here, watch.
Focusing only on the crawling immune cells, we've noticed two classes of them. One was not hungry at all, but
not hungry at all.
Cellular world.
This one here scoops up all this stuff.
Cells scooping up sugars in the ear.
Okay, what we just witnessed was bacteria in this layer here scooping up enzymes, ribosomes, and delivering out through this layer with their little flagellas. Zip, and they were moving fast. That was real time. There was nothing sped up or slowed down. Okay, this is from 2016, before they really recognized this particular layer as this fluid-filled highway, but basically they sort of knew it was there. Now, okay, so let's start to pay attention to this as okay, as I claimed, the fluid-filled highway, which they agree with, it does exist, and we just saw a bacteria going in and out of that fluid-filled highway, and that's the bacteria. These are single-celled. It's the tiniest thing you could ever imagine, and it was living down in these areas down in here, and then dispatched through the fluid-filled highway to wherever something's trying to attack you. And what would trying to attack you? Something that might cause cancer. What does cancer look like? Remember what this is. What does cancer actually look like? Cancer is a tumor, basically. And cancer is an invasion. It's basically an invasion. So it's trying to break through your defensive walls into where it creates all kinds of nastiness and pus and black and who knows what. But it starts to invade. Stage 1, stage 2, stage 3. Stage 4 is when it breaks through the fluid-filled highway. All right? Well, this is actually the fluid-filled highway. They don't realize it. They think it's the same tissue as this, but it's not. They show it that it only takes one little focal point of invasion to get through, and that's what it is. It's a weak junction. It breaks open a couple of cells and forces its way through into your good tissue. That's called a weak cellular junction. In your lungs, they're supposed to be tight, so they can move all around and do all this and flex and everything, but they're tight, so that nothing can get through there. But once they get loose and floppy, you're predisposed to invasion by whatever you're inhaling. I don't care what it is. And the same thing in your digestive system. Every single cell has got a Slurpee around it called small leucine-rich proteins. They're all floppy, gooey stuff, so nothing can get through. But it can float and move around and stretch and do all this. They're gooey substances. Once the cancer breaks through that, then you're in trouble. Now it's eating you. That's what a tumor is. And there's like, right now, I think the last I heard was there's over 400 types of cancers. That means there's 400 types of membranes. In my mind, because it broke through the membrane. The membrane's supposed to stop it. So every one of those has its own chemistry. And in your nervous system, every fiber has its own chemistry that coats it to insulate it. So the chemistry in your body is just phenomenally unbelievable. Now let me show you what bacteria versus enzymes versus chemicals is all about. All right, the whole reason I'm doing this presentation is because of this bacterial study we're going to try to do. What we need to do is be able to collect specimens, so we need some kind of an easy collection kit. We need some kind of an easy exam to examine these collections of what they represent as far as bacterial communities go. We don't necessarily have to know the names of them. We just have to know the spectrum colors, I think. This may work. Be able to look at all of the different molecules in a spectrum and using some artificial intelligence, group it all together and say there's so many of these blue frequencies, because everything's going to have a frequency. All right, every single molecule there is will give off a frequency. So that's what I'm hoping we can use spectral analysis. But anyway, that's all. I think that can be done. And make it quick and simple. Just take a picture of the fecal swab and the oral swab. And we have to get a database of whatever we can get for bacteria. Fecal, mouth, belly buttons, armpits, nasal cavities, wherever. So we need as many details as we can get, but not any personal details. We just need to know the age, the race even, the general demographic area that they live in, any kind of health habits, a few things. The more detail we can get about the person's lifestyle, the more information we'll have about how it may or may not damage or help his DNA through these enzymes. And it's all about the chemistry. You don't have the right chemistry. And I'm going to show you right now, bacteria versus enzymes versus chemicals. Bacteria are the factories. Bacteria are the factories that produce enzymes. Enzymes is the key. I'm going to show you right now. It will blow your mind. But anyway, we need all this, and then we need to just set up a database. This is no medical intervention whatsoever to anybody. This is just collecting samples, doing a study. All right, so don't forget, bacteria are the factories that produce enzymes. I believe I showed you that little bacteria running around with all those little enzymes in his body, the ribosomes, and going out to deliver them on a fluid-filled highway. They also consume waste. They eat waste and bring it down to the lymph nodes to dump it off. All right, listen to this carefully, because this can be taken in a couple of different ways. Enzymes are not alive, and they are not. They don't reproduce. So they're complex chemicals produced by bacteria. The bacteria produce the enzymes. Very complex. I'll show you in a minute. They cannot reproduce, and they cannot actually consume waste. They are makers and breakers of other molecules. They speed up chemical reactions just as unbelievably fast, but they don't get used up themselves. When they speed it up, it's like a complex chemical like this, and I mean basically just about like that. And it comes up, it finds something that wants to destroy or break, it goes click, and instantaneously done. And the other, it's all done, which would take years if you didn't have this catalyst, this enzyme. So these enzymes are ultra important. So they don't even get used up. They go click, 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 and they just keep wiping out all the stuff that's there that it wants to attack, that's been told to attack, or it's programmed to break down certain fats or proteins or carbohydrates or whatever they're made to do. In their chemical signature, they will do, and they don't get used up. So it speeds up a chemical reaction without getting used themselves. They just keep going, boom, 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 boom. However, enzymes are all proteins. All right, listen to this carefully. They're all proteins. Proteins are these polypeptides I will show you in a minute. Some enzymes attack proteins. They're all proteins. But some enzymes attack proteins, so they attack each other. Therefore, enzymes' usefulness is limited by digestion from other enzymes. This is where it's very confusing. What they're saying is one enzyme can be destroyed by another enzyme, so its usefulness is limited by what else is in your body that's going to attack that enzyme, which is a virus. It attacks that enzyme, kills it, so you can't produce the chemistry you need. So now you cannot produce that level of that layer of interstitium that has the right chemistry in it, which I'm going to show you right now, to attack what's coming into it. All right, so I'll show you right now. All right, remember a while back there, Gokul from UC Berkeley was talking about your textbooks are wrong and so forth, and he was showing these little bacteria, I believe he was showing bacteria in the immune cells floating around through there, gobbling up all these ribosomes. All right, these are little balls, the little blue balls it had in its stomach. Basically, that's what I believe it is. Now, these are single-celled bacteria, just tiniest thing you could ever imagine. But they have that little whippy flagella, most of them, which is send them sailing through the fluid-filled highway with this contents. Now, what happens with that contents when it needs to be used? It looked to me like that one we were seeing in Go Calls was it went in and right out. So it looked like it came in there with a purpose to gather all these things up because it was being invaded by something. Or somebody ate something that it had to go get some stuff to break down the carbohydrates or fats or whatever it was. And that's what these enzymes do, because instantly they convert things completely, totally, wipe them out and start them all over again. I mean, instantly. Quarter of a second. All right, now, this is the ribosomes, which are those little blue balls I just showed you a minute ago. All right, you see ribosome? These little blue balls. Apparently, it squirts them out wherever they're needed. And then they unsheath, because they're wrapped in what they call a capsid. Now, I went to Johns Hopkins for genomics, so I know a little bit about this. Marguerite, who I'm working with, it went to Harvard specifically for this interaction right here, the ribosome exchange. And Andy Marino owns a company that specializes in bacterial laboratory equipment, and they set them up for big places where they process a lot of food to make sure that everything is clean and so forth. So, and Andy's, is a triple PhD in bacterial engineering. So we do have some qualifications here. I know I mentioned it before, but it's good to have somebody talking, but you better have somebody that has some evidence to support what they're saying. And here is my evidence. So here is the ribosomes. They unsheath. Then they form these elegant molecules called enzymes or proteins. They're the same thing. Before I told you, it said proteins. All enzymes are proteins, all of them. So what does that mean? There are two things, not just one. An enzyme breaks things. A protein makes things. Proteins are building blocks. Enzymes are breakers. That's the difference. All right, now don't forget, this is going to turn into an enzyme from that ribosome. And this is what the enzyme, you saw this. And here's what the enzyme really looks like once it unfolds. It's just like this. Now, every one of these is a magnetic signature. Enzymes are so elegant that they actually do click chemistry. So if they come up against something they don't like, say this guy here says, this thing shouldn't be here, I'm going to kill it. It comes up and goes click, done. Instantaneously, brrr, case is closed for that thing. It's all done at the speed of light, literally, which was the speed of magnetism. Bump, 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 which is, I call it click, 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 click. Click chemistry is instantaneous enzyme destruction. All right, or it may go click and set up a whole series of building blocks. And then those building blocks incorporate into each other, and they make fibers and all these kinds, because their building blocks are collagens and keragens primarily, is what I'm talking about. All right, just so you understand, there's a zillion, all kinds of thousands and thousands of different bacteria, and they show them in these different shapes and arrangements and so forth. But the reason they're like this is to create these magnetic signatures. This one here is going to create two balls like that, two magnetic balls. And that one there is just going to have one with a seam in the middle, and that one there is just going to be one big ball. But they're all going to be magnetic particles that create these extremely elegant enzymes, which are created from the bacteria. These are only single cells. Bacteria is one cell, tiny. All right, and then it spits out that ribosome, unsheaths into the enzyme and the proteins. Now, let's just look in a nutshell exactly what happens from the bacteria to our DNA. That's a sequence we want to go over right now. All right, this is it in a nutshell. The bacteria itself has its own DNA, just like we have. And inside its own DNA, it says, I am going to have a flagella, and I am going to have a certain membrane around myself, and I am going to create ribosomes. And those ribosomes are going to be strung out in this particular exact sequence of these amino acids, which are the protein chain polypeptides, just like this. It spits them out in a chain. All right, because it's got a program. It says, all right, add another one of these over here, and another one, and the blue ones, a green one, and they come out in this protein chain. Now, it goes into this section right here, which is where it transfers this chain of sequence into this chain of messenger RNA. So the transfer RNA changes this messenger RNA, sends it out, and it breaks in to our DNA and incorporates itself here. But it requires this polymerase. All right, polymerase. And what is polymerase? Anytime you see ACE on something, it means it's an enzyme, basically. This is just nothing more than an enzyme. If that enzyme's not functioning correctly, you're not going to be able to get that messenger RNA into your own DNA. And again, this is an enzyme. An enzyme requires a bacteria. I don't care what you say. That's just the way it works. If you don't have the bacteria, you're not going to have the enzyme. And an enzyme is required for the messenger RNA to do its job. Simple as that. And the same thing with this little cloud here. You have to have the right transferase. I believe it's called transferase between the polypeptide chain and the messenger RNA. It transfers that code, and then it goes and invades our DNA. Now, this could be a good guy, it could be a bad guy. That's the key. If it's a bad guy, it comes through here, and then it attacks us in a number of different ways. It could invade our DNA and just change our DNA. I don't know. Or it may not invade our DNA, it may not be able to get that deep into us, but it might be able to take up residence with its own set of molecules that grow. Because I don't know what's coming out of these bacteria, and when you take them out of the wild and so forth, from animals that developed them, bacteria has to evolve. This is what evolves. It's not us. It's the bacteria. We're products of bacteria. I'm not kidding you. That's a fact. If you don't have the bacteria, you don't have the enzymes. So whatever bacteria is in you has to produce these enzymes. Now, these enzymes are good or they're bad, both. A virus and an enzyme that's a good enzyme is the exact same thing. So you need enough good enzymes to fight off the bad enzymes. And you should easily be able to do that if your immune system's working, because you have a fluid-filled bag. Every single molecule in your body is touching this fluid-filled highway of enzymes that should protect you. But if you don't have the bacteria that is good for you, none of this happens. None of that. So you could be forever sick because you can never program yourself to recognize and fight back. That's what this whole thing is about becoming immune, is to be able to reprogram the DNA. But in order to break in, you need an enzyme. That's why I'm really pushing so hard on a bacteria database. If we don't have that, we're stuck. We don't have any idea what we're doing until we have a database. And then we can associate patterns. This is what the whole thing is about. Okay, for years now, I have been talking about nucleophilic substitution. Here's what's going on. Proteins and nucleic acids are methylating enzymes. Wait a second. All right, I think I talked about this before, but if you do not have the correct enzymes to turn on methylation, all right, protein and nucleic acid methylation enzymes, methylating enzymes, are implicated in myriad cellular processes. The enzymes utilize diverse chemical mechanisms ranging from nucleophilic substitution, that's the most important one, which is displacement to a novel radical-based reaction found in bacterial iron, sulfur cluster proteins. Now, it goes on here to talk about they work with your DNA. All right, enzymes, they establish, recognize, and remove DNA methylation are broken into three classes: writers, erasers, and readers. So we're writing into the DNA, we're erasing out of the DNA, or we're reading from the DNA. That's what the methylation does. Now, if you don't have the enzymes establish, recognize, and remove DNA methylation. So the enzymes come in and they take care of DNA methylation. They're broken into three classes. They can come in and work as writers, they can come in and work as erasers, or they can come in and work as readers. Or they cannot come in at all because you don't have the enzyme. All right, or you may have a weak enzyme and your DNA methylation is slow or erratic. All right, but DNA methylation means you turn on the signal or you turn it off. It doesn't matter if the gene's good or not. If you can't have the methylation happen, it really doesn't turn on. It doesn't do anything. All right, I don't mean to be so detailed about this, but it's all in the details. This is a word called allosteric, and it means relating to or denoting the alteration of the activity of a protein through the binding of an effector molecule. It means that the molecule you're putting on affects the reaction at that specific site. So we're altering the activity of a protein. All right? This is right here. Listen to this. This is about protein and nucleic acid methylating enzymes. These are about the enzymes. If you don't have them, you don't methylate. You don't methylate, you don't turn on. This is mechanisms and regulations, and here it is right here. Let me come to a point where it makes sense, so I'll make it quick. Okay, real quickie here about methylation. National Library of Medicine, National Institute of Health. Cut right to the chase. Recent advances and emerging trends in the study of methylating enzymes. These are the enzymes that are made by bacteria. So the study of these methylating enzyme mechanisms and regulation highlight the importance of protein and nucleic acid methylation in cellular physiology and disease. All right, so this is National Institute of Health, not me, talking about how important protein methylation is. And the enzymes that control it. And the only way you get the enzymes is from the bacteria. Okay, here's another area I think needs a little more interest. Epithelial cell junctions. These are the cells that coat your surface of your body so that you can't get penetrated. Now, there are different three are different types of, it means there are different types of connecting junctions that bind the cells together. Okay, I've been talking about this a lot because I think this is what creates pre-existing conditions. Now, there are four main types of cell-to-cell junctions. All right? There's different types of connecting junctions that bind cells together. So all cells are supposed to be glued together as if they're a membrane so that you can't get through them. They got to be glued together with these junctions. Now there's occluding junctions, which are tight junctions. Your lungs are supposed to be tight junctions. As you breathe in, you're not supposed to let these things squeak through these junctions into your good flesh. And that's what I showed you, what happens with cancer. All it's got to do is find an easy entrance, like this, and it breaks through and gets through. There's no enzymes here to protect you. All right, that's my take on that. All right, so there's let's go with four of them. There's gap junctions, desmonas, adhering junctions, and some of them they can move and all this stuff. Like your lungs have to do all kinds of stuff, but they have to have very tight junctions inside the alveoli because you're taking in all kinds of who knows what. And some people get sick from inhaling it, some don't. And some people get sick from inhaling other people's bacteria, and some don't. And why? Because some of them have tight junctions where they're supposed to be tight. Some of them have loose junctions. The loose junctions, they open up and straight through. All right, these are the junctions. Now, adhering junctions are right like this here, and they should be together, really tight. And this actin filaments, which are microfilaments shown in red, join up to adhesion junctions. So they adhere together. Now, if those actin filaments aren't, there's supposed to be tons of them in your body, just everywhere. And if you don't have those filaments good, or they're weak, or there's something wrong with them, or those filaments are not, they don't tangle the way they should tangle, you're going to have loose junctions, I would think. This would be something to investigate. I don't know. But I would like to know if people that get lung cancer and so forth, even the non-cancerous tissue, has it got tight or loose junctions? Is it something that was just a thing just waiting to happen because there wasn't a tight junction? That would be interesting to know. And that could be done through autopsies and some real fancy microscopy and that type of thing. But all of these different types of junctions need to be looked at in a new light, because this is how you're getting invaded. In my mind, it's got to be coming through that junction, because that junction is your membrane, and your membrane is your, basically to me now, it's your immune system for that particular organ, or that particular muscle, or tendon, or eyeball, or ear, or gums in your esophagus, all your intestines, all through your guts. Even your red blood cells have membranes, which have membranes just like that around them, right around your red blood cells. And if there's something wrong, you don't have the right protective chemistry in there, you get some kind of blood disease. That's the way I am seeing this now in the research that we've been working on, and we're hoping that will establish itself as possibly factual if we can get this database, and then we can make some correlation between what happens to people, and is there some biological bacterial issue without the enzymes. And can we intervene? And is it demographic? Is it everybody in that area? Because it appears to be that there's a lot of congested areas of certain diseases. Why? Not 100% sure. But we should be able to track and see, is bacteria being damaged in areas that have like these big train wrecks and all that stuff? Is that causing people's bacteria to be damaged? Because I believe if the bacteria gets damaged, and it's everything you take in is going to affect you. If you can protect yourself from it, I suppose you're going to be okay. But some of that stuff you probably can't protect yourself from. It's just too toxic, and it gets right down through. And then, but I would think it's going to affect the bacteria. We got to look at the bacteria and get a bacteria database, because right now it's a lot of guesses. A lot of guesses at this particular moment. But anyway. Okay, this just came out talking about brain acidity linked with multiple neurological disorders. Well, what causes the acidity? Well, it's chemistry. Now, if my brain doesn't have a lot of acid on it and yours does, what's the difference? How did that happen? Here's the deal. They all involve a degree of cognitive impairment. So they're all like Alzheimer's, autism, all of these things that affect your nervous system. They often share genetic or metabolic features, hinting at a common biological basis. Well, metabolical features, what are those? That's digestion. All right? Genetic is usually within the same family. But usually within the same family, they're eating the same foods, they're drinking the same water and all that same stuff too. A lot of that can happen as a result of being exposed to the same things, possibly. Who knows? But metabolic, that tells me we're talking about digestion. All right, here's what they think. The saying, let's start with the definitions. Digestion, which I just talked about, it's about digestion. Well, digestion, they say, refers to how the body processes the food in a gastrointestinal, which is your GI tract, and how it eliminates the food waste by the intestines. Well, I totally agree with that. Now, but the metabolism happens at the same time. It's all through this digestive tract. So, metabolism, they say, and I guess I have to agree with this, metabolism refers to how the cells utilize the energy we have absorbed from food during digestion. So, if we didn't digest something, we don't have the energy, we don't have the products from the digestion. And the only way we can digest something with that enzymes, the only way we can have the enzymes is have the bacteria. So if your bacteria is damaged, I don't care, all the rest is meaningless, because you're not going to be able to digest, and because you don't have the enzymes. You don't have the enzymes, you don't get the chemistry. You don't get the chemistry, it leads to some kind of a condition. Or loose junctions. It could be just something moderate, very, very low impact, but it could be something just absolutely terrible. And if all your junctions that are supposed to be tight are loose, that's not good, because then you're just a wide open case getting ready to be invaded. Let me just read you this one. All right. Every cell has a bilipid membrane. Hold on. Okay, once again, no medical advice whatsoever. This again, National Library of Medicine, National Institute of Health. They're talking about anti-tumor effects of fecal microbiota transplantation, implications for microbiome modulation in cancer treatment. So these fecal matter transplants put the bacteria back into somebody who is deficient in some form or another with bacteria, or they have too much bacteria. And in some cases, what they do with these fecal matter transplants is they flush them with all kinds of antibacteria, antibiotics. It kills them all. Zip, wipes your whole body out. And normally you would just die after that if you can't reestablish your bacteria. And for a long time, they were giving people antibiotics. I had to have surgery twice because I took antibiotics for a root canal, and I didn't have any trouble. He just said, just take them, don't worry about it. And I ended up I had a colonoscopy, a colon repair twice from eating through the walls. If you don't have the right bacteria in there, you can't protect yourself. So anyway, this is about fecal matter transplant from healthy donors, one of the techniques for restoration of a bad gut, dysbiosis. It's increasingly being used to treat various diseases. Notably, mounting evidence in recent years revealed FMT, which is this fecal matter, has made a breakthrough in oncology treatment area. It's cancer and all kinds of stuff they're finding now. That's why we want to get a database. Now, this is just from 2022. And believe me, this stuff will travel so slowly because everybody's scared of getting sued. So they don't want to even think about this, because once they do, and a patient, because I don't want to go into it. Believe me, it's very, very hard to get anybody to pay any attention to this. But they have been doing this, looking at this, and it's been going on forever. The Chinese were doing it for thousands of years, literally. All right, the gut microbiota target for therapy. That's because you need those enzymes. You see this? Even Yale University, eat, poop, and live. They know that it's been going on throughout history. Dogs eat poop. All animals eat poop. And they feed their poop to the babies when they're young to get their bacteria going. This is 2016. Eat, poop, and live, Yale University. They say, yeah, we know all about this stuff. Well, why don't they use it? Because it's against the law to have a fecal matter transplant. That's why I'd like to see the research prove that the bacterial issues are the problems, and then how do we address that? How do we infuse the bacteria? Maybe we don't have to use fecal matter transplant. Maybe you have something else. But this is the research that needs to be done. That's all. Okay, this is Harvard Medical School talking about stool transplants are now standard care for recurrent C. difficile infections. C. difficile was a deadly disease, and they have a certain regime they want to give you that's, drugs. But a lot of people, they just couldn't stop it because they didn't have the bacteria to create the enzymes. So they gave them fecal matter transplant, and boom, virtually all of them came around. Very, very high percentage of, look at about 20% of people treated with antibiotics will develop recurrent infections, and the best way of clearing recurrent CDI is with FMT. And this is Harvard. This is not me. Not me. I'm not saying any of this stuff. That's Harvard. And what we want to do is make a database of the bacteria so we can make these assessments of who's susceptible to what. If they don't have the right bacteria, and you see everybody that has that bacteria is healthy, and this guy doesn't have it, and he's got all kind of issues, well, let's investigate what affects what. And you develop patterns. That's just the nature of research. All right, here's one. This is specifically what we were originally looking into, was emotional disorders and autism and even Alzheimer's and Parkinson's, all that stuff. How it is affected by the bacteria. Now, this is again, National Institute of Health. No wacky, crazy people. This is National Institute of Health. Effect of fecal microbiota transplant in children with autism spectrum disorder. A systematic review. So they systematically are trying to figure out what this is. Can it work? Can it help? And this is just from 2023. And again, very slow to work with this stuff. And the results were, the conclusion was, compared with the baseline, fecal matter transplant significantly improved symptoms of autism in children with autism spectrum disorder. So it's a spectrum just means that they can be really just talking and absolutely banging their heads and biting and screaming, all the way to just a mild difference in the way they see things. Let's put it that way. It's a spectrum. Now, so fecal matter transplant improved symptoms of autistic children on this spectrum in observational studies. They just watched them, and this is what we had done in Northern Ireland with Marguerite's son, John. However, rigorously designed randomized control are needed to establish the safety and efficacy. You hear this compared? It's improved symptoms in children in observational studies. Well, let's get it going. Let's get that study going, I would think. And then it goes on to all about how they did it and how they did the study and everything. But the bottom line is it worked, and it significantly affected. Let's see if there's a. Well, it's all here in the study. It significantly helped. All right, here's again. National Institute of Health, not crazy people. Fecal microbiota transplant role in the treatment of Alzheimer's disease. A systematic review. And it's the same thing as they did with the autism study. It was a systematic review. But they get to the end and they said, well, it looks like everything was going good. It looks like it's helping, but there's just not enough studies to give it a conclusion to say, yes, it's good. So we don't really recommend it because they didn't do any studies. And this is about Alzheimer's. Now, and they're doing the studies. This is an Alzheimer's neurodegenerative disease. Starts slowly, worsens progressively. And that's exactly what happens when your bacteria is not working. Now, they get into the bacteria and the gut dysbiosis now because they're starting to realize. And at the very end, they actually say, yes, listen to this. All right, at the very end, I go to the conclusions. I mean, I look through everything, but the conclusion is what makes the whole deal. Here's the conclusions. In conclusion, fecal matter transplant can potentially become one of the modalities in treating autism. Autism and Alzheimer's, exerting its effect through the microbiota gut-brain axis. Microbiota. And it affects your brain because it's affecting the chemistry. It controls your brain. Now, the diversity of the gut microbiota in these patients is widely changed. So they have widely changed gut bacteria, totally different from healthy people. Healthy populations as the type of bacterial abundant in healthy people, differing from those in this population that have this Alzheimer's disease. It's speculated to participate in, they think that the gut bacteria participates in this because the original gut bacteria metabolize peptides, soluble fibers, and dietary proteins, bringing out these different chemicals and tryptophans along with other metabolites. It's basically you need the enzymes. These products work to lessen gut inflammation. The gut inflammations happen because you don't have the protective chemicals in there because you don't have the bacteria. That's my position right now. And it gives permeability. All right, and that's with the blood-brain barrier, BBB. So it works to lessen gut inflammation along with blood-brain barrier permeability. All right. Animal studies have demonstrated fecal matter transplant effect to restore these different chemistries and a healthy microbiome to disrupt the oligomers, decreasing Alzheimer's disease pathogenesis. It doesn't keep going bad. And after 6 months, they say it starts reversing. However, the lack of clinical trials, once again, lack of clinical trials due to ethical concerns. The ethical concerns is they should try to figure out what's wrong with these people. Hindered our findings in humans. Lack of trials. Nevertheless, 2 cases were identified in an 82 and a 90-year-old patient that had received fecal matter transplants from fit donors, and the recipients exhibited cognitive improvements. Future recommendations from this review would be to conduct further studies, especially randomized controls, where fecal matter transplants are implemented in patients of Alzheimer's disease at various stages of the disease, following them up for longer durations. Thus, a further detailed assessment of its effect as a possible Alzheimer's disease intervention could be evaluated. All right, so it's always, well, we need to look more. We need to look at it. So, yeah, we don't have enough data. We really need to start looking into that. That's a couple of years ago. So, and they knew this for many, many years now. This is not something that was unknown. It's just not taken into consideration by the medical industry. I hate to think of why, if it's got related to money. I don't know. I find that hard to believe that people would. Anyway, but we need to start looking down these roads that I'm talking about, and it's every molecule in your body has to be serviced instantly, over and over and over, 24 hours a day. So the enzymes have to keep doing their job 24 hours a day. That means your bacteria has to be very healthy. So you also have to feed that bacteria. You have to put them in an environment where they're not being damaged. And we're eating so many bad things. And then in the sunlight, you're burning through your skin, and actually the bacteria that's in your skin is being killed. And then you get melanomas because the bacteria that's in the cells that are supposed to clean that stuff out they're not alive as they should be. So you start to develop melanomas. And that's something else that shows promise with this fecal matter. Let me show you that. All right, I just talked about melanoma. Listen to this. Dietary tryptophan metabolite released by intratumor Lactobacillus reuteri. It facilitates immune checkpoint inhibitor treatment. So it helps in this immune treatment of melanoma, primarily is the one I'm interested in. Now it says the use of probiotics by cancer patients is increasing, including among those undergoing immune checkpoint therapy. Here we elucidate a critical microbial host crosstalk between probiotic released hydrocarbon receptors against these invaders, basically, within the tumor and microenvironment, potentially enhance anti-tumor immunity. All right, now don't forget, this is for melanoma. Okay, this is from the National Institute of Health again. This is 4 years ago. They're talking about Lactobacillus reuteri, the prevention and effect on melanoma. Now, this is what happens, is this Lactobacillus reuteri apparently creates this TNF-A and TNF-Y, which is tumor necrosis factor. And it says, on further examination, it revealed the levels of these are enzymes, in the serum were significantly increased. In summary, it was speculated this L. reuteri could stimulate the body to produce anti-cancer cytokines. Well, it's producing chemistry, yes, absolutely. It can inhibit the migration and colonization of melanoma cells so as to prevent the onset of melanoma and prolong the survival of mice. That's what it says. This is not me. The experiment was a useful complement to functional studies of probiotics and provided theoretical data support for development of new microbiologics. Now that's 4 years ago. But it is, again, this is not just off the wall science here. This is people that do this and they report on these things. But it does not filter through the medical community, I don't feel, as the way it should. And that's why I believe research is what we need. And again, that's all I do is research. I don't recommend you. I don't say take this stuff. No, absolutely not. It could hurt you as far as I know. That's why we need to find a database. Because they say these things, and I look at it and I say, oh yeah, go out and take a bunch of this stuff. No, absolutely not. I don't know if it'll hurt you or help you. But it does something. They say when people are taking this, they get this tumor necrosis factor increase. Is that right or is that wrong? That's what we need a database for. All right, to find out what bacteria is in you and what's in me and what's healthy and what's not healthy. And that's what this whole presentation today was about. Now, I know it took a lot longer than I had suspected it would, but you have to know all the ins and outs of this to understand how important this is. And it's staggeringly important. And it's mostly for the kids. This stuff about the autism and all that, Marguerite, who is in Belfast in Ireland, North Ireland, she's now in the news now. They're following her story through the news over there in North Ireland, and how she got started and how she developed this regime for her son, John Jr. And again, we're going to have a meeting actually today at a couple of hours online with the people over in European Union, because it's taken over there. They have a lot of problems. We do too. Everybody's got kids or somebody they know with autism. It's just 100%. It's right across the board, and it's getting worse. And the only reason I think it's getting worse is because of the environmental conditions are wiping out the bacteria in the body. That's what it seems to be looking like to me now. So it's just going to require a little bit more research. and that's going to probably require some money. I don't want any money, and I'm not going to do the research either. I'm just a guy talking, but I have a bunch of people. I have a lot of people that I've been working with, a lot of people that have a lot of expertise, but they have no sway in the community, and I don't either. And no matter what it seems that you can present, it's very, very hard to get an audience. And even though Andy's like a triple PhD and he's a bacterial engineer, he knows all of how these things fit together and how they work together, and he's got his own business and the whole 9 yards. And I don't think he's getting a whole lot of attention either. But it's hard to start screaming because they come back at you. That's the problem with this whole situation. So hopefully it'll get some attention. If we can get a database, we can make some valid assertions. And then once you know the truth, there is no excuse.